Corona, COVID-19, a new virus hit. I bet you've heard this word like 100 times a day, but are you sure you know everything about it? If you are not, then this video is for you.
New stuffs to know.
Hello everyone, this is Dr. Khalid. Today we're gonna talk about something is going viral, and ironically, it's a virus. It's Corona, of course. And we're gonna cover many aspects of this problem. So let's start. So at the beginning, it's belonged to Corona family, which actually is like zoonotic viruses which mainly infect animals, but sometimes it jumps to humans and cause usually mild problems like flu or mild or moderate respiratory syndromes. But in some cases, it jumps from animals to human, which actually cause a real problem, like the two deadly viruses, SARS, MERS, and the new COVID-19. So why Corona? Corona is a Latin word means a crown, and this family of viruses look like wearing a crown when it's looked at under the microscope. The COVID-19. Its full name is Severe Acute Respiratory Syndrome Coronavirus Related to, or COVID-19, or as widely known as just Corona. Is composed of protein spikes, lipid envelope, and RNA core. The first case recorded was a 55-year-old man from Wuhan in China. It was on 17th of November 2019, and now after like 5 months there's over 1 million cases worldwide. Frankly, despite the fact that the virus engineering techniques already exist and available, but until now there is no strong evidence or scientific evidence to say that the virus is human-made or just escaped from labs. And before we talk about that, we should know a few things about how do actually viruses jump to us. Actually, virus is considered as a non-living things, and as I've mentioned before, it's very simple in composition, and to be honest, this is one of the most important keys of its success. And for the virus to survive, it needs the 3 stages. First is to find a host, second, to multiply in that host, and the third to infect others but from same species. But while spreading, it may accidentally infect other species. But how's that? Generally, the cells should have specific receptor at its surface in order to allow the virus to invade them. These receptors are unique and differ from species to species, and even from tissue to tissue in the same type, which is determined by the genes of each species. And the genetic dissimilarity usually prevent the viruses from transmitting from one species to another. However, because viruses are very rapid in replication and can make millions of copies within few days, they can easily develop a random mutation in their genes. Because of the general rule, the faster the replication rate, the more chance of mutation. In fact, most of these random mutations have no effect or even cause inactivation of the virus, but sometimes in small proportions when these changes find the right formula in how to attach to a new type of cells and replicate inside of them. Then it enables the virus to jump and infect a new species. And there are 2 major factors that influence the successful mutation. The first is how close the genetic material between the previous and the new host, because the closest, the more chance to have a successful mutation. The other is number of infected species in a specific area. As it raises, it increases the odds of getting that mutation. So, most of, if not all, the deadly human viruses came from non-human origin. And there are many examples, such as a bird flu from birds, swine flu from pigs, Spanish flu from chickens, Ebola virus which came from bats, HIV from chimpanzees, and 2 of the same corona family, SARS which thought to be came from civet cats, and MERS from camels. Some of the evidences suggest that it's originated in bats, then transmitted to pangolin, which in turn it delivers it to us. And the pangolin is scaly types of anteaters, and the scales used in traditional Chinese medicine,
hence on the red list of threatened species.
But wait a minute. To make a successful transmission, all the hosts should be in frequent contact, right? Yes, of course. And here where Wuhan takes place.
When the infected cases traced down, they found that 27 of the first 41 cases, including the first case, was in contact with the same wet market in Wuhan, the same markets type that provoked the SARS virus in 2002. And wet markets are places where living dead animals are overcrowded and in constant close contact. As you can imagine, the large numbers of animals in cages and negligence and how the flies of the sick animals came in contact with the good ones and then to humans. And this happens on an everyday basis. So there's exactly where the perfect environment was met, where all the three hosts are always available and in continuous contact.
China is a large country with a huge population and a wide variety of animals on food menus, including wild ones. And each one of those animals has its own type of viruses, which could cross-spread to other species when they kept in a close contact, exactly like what's happening in that type of markets. And the story behind that large variety of those markets started at the 70s, when the government was controlling all types of animal resources, and even at that time, population was so large. And with the failure of the government to feed the people at that time, so a famine was happened and killed about 30 million people. Although the government forced to give up the control of resources and allowed the private farming, but that didn't solve the problem because farming got controlled by large companies that pushed the small farms to catch wild and unusual animals like reptiles, snakes, turtles, and bats, and others, and used them as a food to sustain themselves. Then eventually, the government legislated to encourage those people to continue their activities, to keep them out of poverty, which led to the born and the expansion of this industry from the small farms in China. However, this also opened the door for farming and trading in illegal and threatened types of animals as tigers, rhinoceros, as well as the pangolin. When an infected individual coughs or sneezes, he spread his viruses in his droplets, and whenever the healthy person came in contact with those droplets, they entered his eyes, nose, or mouth, the virus will enter his body. First thing, the virus will search for a specific type of cells that contain the specific receptor on their surfaces. It's called the AC2 receptor, which could be found in mouth and tongue, but in a larger amount in the lungs. So when the virus finds those receptors on the respiratory epithelial cells, it will bind to those cells by its spikes. After binding, it will mix its genetic material with the original genetic material of the host cell, and subsequently it will control that cell to produce copies of itself, so the cell function will be lost, and the cell will turn to virus factory which will produce new viruses, then the new copies will invade new cells and so on. This could make about 10,000 copies within hours and millions of copies within days. However, the problem didn't start yet. Now the spreading of the viruses throughout the body will release the inflammatory mediators, mainly the cytokines, which in turn cause the immune cells like neutrophils and T killer cells to fear. These cells would overreact because they didn't recognize the virus. Then a battlefield would happen, which will end up by destroying the virus, but with remarkable damage to the host tissue and even the gut cells. Okay, now the real problem shows up, that the battlefield happened at the lung, and the damaged tissue is the epithelial cells of the alveoli of the lung, which is the functional unit of the respiratory system, and replaced by fibrosis or scar tissue. This actually leads into two big problems. The first is the fibrosis and fluid from the remnants of the body resistant will prevent the lung from functioning normally, which will lead to respiratory failure, so the patient needs a ventilator at this stage.
The second problem is that the bacillus left the lung without its main protection layer, and means the lung epithelial cells, which will allow the opportunistic bacteria to infect the lung and causes pneumonia and eventually blood sepsis, adding to that the existing respiratory failure, which unfortunately ends in death of the patient. But this is not how it goes always. The death consequence happens only in 45% of affected people who are usually over 60 years, immunocompromised, or have a pre-existing respiratory conditions or other medical conditions such as diabetes, hypertension, or heart disease. While in other individuals, the immune system usually succeeds to destroy the viruses and the infected cells only with a minor lung tissue damage, so everything goes just fine. The most common symptoms are tiredness, fever, and severe dry cough, and some patients show flu-like symptoms as generalized ache, sore throat, nasal congestion, or even diarrhea. However, some patients got infected but experienced no symptoms at all, but the problem is they're still able to infect other vulnerable people. Scientists sum up how infectious diseases spread with a number called basic reproduction number, or simply R naught. And this indicates the number of new cases who are gathered directly infected by one case in the population. So R naught of 2 means that the infected person will infect 2 other people, and each one of the 2 will infect another 2 persons, and so on. In general, R naught less than 1 means that the disease will disappear in the long run, while R naught larger than 1 indicates that the disease is contagious and it will spread out. And the formula of calculation considers all environmental and biological factors. For example, R naught of measles is between 12 and 18, and that shows a high infectivity, while Ebola virus is 1.5 to 2.5, and for the flu is 1.2 to 2, and for COVID-19 is estimated to be between 2 and 2.5, which of course higher than flu, but at least it's less than measles. According to studies, less than 2% of COVID-19 cases was in children age group in China. And the same thing shown by studies done on the mice during the outbreak of its cousin, the SARS, which showed that the children are rarely infected but poses no symptoms or have a least chance to develop serious consequences. Actually, the real reason is not clearly known, but there are theories which could explain that. Generally, as a human getting older, the lung lose their ability to fight viral infections because of the aged immune system, so it become mature but weak. Also, children have more active T cells than adults, and these type of cells are needed to resist viral infection. Also, it regulates the inflammatory response in addition to the immature immune system of the children. All of that help and diminish the damage that happens to the lung, because as we know, the problem isn't in the virus, it's in our immune response. Another hypothesis is that children have more ACE2 receptors. Yes, the same receptors which used by the virus to enter the cell, as the larger number of these receptors means large amount of the ACE2 enzyme important role in immune regulation. So yes, the older have much lesser amount of it, so it may limit the number of viruses which enter the cells, but it will increase the bad immune response of their bodies, so that means more tissue damage. One exit theory: there are 4 other weak viruses from the same corona family infect the children because of their immature immune system, and all of them cause symptoms similar to common cold, so when they get COVID-19, their bodies won't be much surprised. And all of that is just theories about it, but we still don't know how exactly this happens. Scientists test the virus in different surfaces and environmental situations, and they came up into that on average the virus can survive in aerosol droplets for 3 hours, and on cover for about 4 hours, cardboard about 24 hours, 4 days on wooden surfaces, and for up to 5 days on ceramic, plastic, and stainless steel surfaces. But all of the detection done with the PCR technique
which detect the viable virus. But we don't know for sure how much viable virus is needed to infect another healthy person. The investigation done by a technique called PCR, which stands for polymerase chain reaction, and the first step is by obtaining a swab from suspected person. These swabs usually taken from patient's nose or mouth, then it put into machine that duplicate the genetic material. So if the virus exists, the produced copies will amplify its footprint, so we can detect it easily by other antigen antibodies techniques. However, some errors could happen, like what happened in Wuhan. Actually, many people tested negative, but unfortunately they turned out to be positive cases and infect other people. And this depends on which stage in investigation you are. And this could start at the swabbing stage, which may present the first stage of error, because usually it's taken from mouth, and COVID-19 is a lung pathogen, so if it's not spread widely enough, the results will be falsely negative. The second stage of error is carrying time, because it takes time and the proper conditions are not always guaranteed. And the third is a human error at labs, like if they are not well trained to use the test kits. Before we talk about that, let's know how does vaccines work. At the beginning, when any pathogen gets into the body, it will trigger the immune response, like macrophage and the B and T cells. And when it destroys the pathogen, it will store info about it, which will help to defeat it if it appears again. But this usually takes time, which allows the virus to spread out. So if the immune system was not strong enough and couldn't handle the attack, illness develops and sometimes death. So here where the vaccine rule comes. When you got vaccinated, the macrophage will engulf it and allow the T cells to recognize the antigen or the ID of the pathogen. Then it will produce antibodies and a special type of immune cells called memory cells. And those will stay long in your blood and immediately recognize the real pathogen once it got in your body. Then helps to destroy it faster. So it trains your body in case of that pathogen really attacks you. And this actually works for bacteria and viruses. And the vaccines usually two types: live vaccines and killed or inactivated vaccines. And there's a relatively new type which is called a subunit vaccine, which actually takes a part from a virus and make it as a vaccine. And this is the case in COVID-19. The scientists took a part of it, which is its spikes actually, and make it a vaccine, so your body will readily recognize and destroy the virus once it enters your body. To release the vaccine to the market, we need to pass many stages, and those steps should be granted. First, safety, because some vaccines are effective but it may cause strong adverse effects, such as what happens in 2004 when they produced a vaccine for SARS, but it turns out that it caused a real lung damage when the patients got the virus. The second is the efficiency, as of course we don't want a non-effective vaccine. And the third is the expansion, as it could cover the market demands. Also, the time it takes to isolate the virus, gets its genome to make a vaccine, then test it the vaccine in vitro, animals like mice, then non-human primates, then finally humans. So imagine how does it needs to get a license for a vaccine and produce it in massive amounts. On the other hand, the majority of vaccines producers are private organizations, and actually they are looking for the financial interest. So it usually takes between 1 to 2 years, but some of the optimistic scientists with taking shortcuts in these pathways suggest that the vaccine will be ready within 8 months. Actually, some studies and trials done to investigate the effectiveness of the antimalarial drug, the hydroxychloroquine, and they sum up that it actually have an antiviral activity, and they show impressive results on treating the COVID-19 cases. But the problem here, actually, the COVID-19, it has a high spontaneous recovery rate, and all of those trials lacking the sufficient number of subjects or don't have a control arm, who are the same people in the same situation but not receiving the treatment, and we use them as a comparison group. So the scientific community cannot rely on these results to license that drug to be used on the COVID-19 treatment, in addition to its serious adverse effect which could be caused when used in the wrong condition. However, there is a large study ongoing now in Europe which involves more than 3,000 patients, and they are trying to figure out if this drug is actually effective or not, besides another huge multinational trial which is study 4 extra potential treatments for the COVID-19. So hopefully they could find it. So as we know now, it's a serious disease, but we can control this and that. And as it became a pandemic, we could make it a slow pandemic by either increasing the health services, which actually sometimes is not applicable because it needs time and money. The other thing is just by washing our hands, because as we've mentioned before, the virus have a lipid envelope which could be destroyed easily by the soap, and following the social distancing and self-isolation measures. Otherwise it will be turned into fast pandemic which will exhaust the health services, and then it's gonna be a real disaster. So it's in our hands, literally and figuratively. If you like this video and you want to see more of this type of videos, don't forget to hit the subscribe button and enable the notification button. And don't miss the chance to be one of the pioneer subscribers of this channel. See you next time.